I'm going to cover axonic transmission in a neuron and the method by which neurotransmitter crosses the synaptic cleft, carrying an action potential from one neuron to the other. For this video you need a basic knowledge of depolarization in a neuron and the difference between an axon and a dendrite. The relevant things will be explained as you go. This is the structure of what we're looking at today. As an overview, so this doesn't look very confusing, we'll start from here. These are vesicle packets, circular, and the red parts inside them are neurotransmitter. This could be dopamine, as we know. This could be epinephrine, other things, we'll get to it. But each neuron is responsible for synthesizing and using one type of neurotransmitter. They use a subset of other minor neurotransmitters as part of their complexity in information transmission. But generally, if it's a dopamine that's dominant here, see the red and the pink, if the dominant one is dopamine, we'll say this is a dopaminergic neuron. If the dominant one is GABA, G-A-B-A, which is the major inhibitory neurotransmitter in the CNS, then we'll call it a GABAergic neuron. Same way, glutamate will be the most major excitatory neurotransmitter, and we'll call this a glutamatergic neuron if it synthesizes and produces that mainly. So if we consider this to be the axon, we know that it's connected to a cell body, a soma, neuron soma. Inside the soma we have a set of structures surrounding the nucleus. We have the endoplasmic reticulum, we have a Golgi apparatus, we have a bunch of mitochondria and some other structures. We're focusing on the endoplasmic reticulum, specifically the ribosome portion which produces neurotransmitter, and the Golgi apparatus which packages neurotransmitter. From the packaging process, the vesicles are sent, it's called a transport vesicle, they're pushed into and sent down this structure we know very well as the axon. Inside the axon being microtubules which actually carry the vesicles down to the axon terminal. This is a process that's independent of the action potential. Common misconception, I was confused by this at first as well, but they are two completely independent processes. The action potential actually travels on the membrane instead of inside the axon, like a wire. It's not like a wire. This is to say that the action potential would travel on the rubber of a wire as opposed to in the copper. So the neurotransmitter actually goes down the microtubules and ends up here before the action potential even gets there, so it's primed and ready to go. Now we'll discuss some issues that we might have with the microtubules in certain diseases in a bit. But let's say the neuron gets depolarized through a summation of charges, ion flow from the dendrites of this presynaptic neuron. Pre because it's before the transmission is passed, and this is a postsynaptic neuron. So when the action potential travels down the membrane, it reaches these receptors, these channels here, responsible for letting in calcium with a 2+ ion, 2+ electrical charge. And once the action potential reaches these ion channels, it draws in calcium 2+ from the environment, and these present vesicles get adhered to the membrane of the presynaptic neuron. Once they are adhered, the neurotransmitter exits into the synaptic cleft through a process called exocytosis. We'll explain the specificities of this later, as we will with the microtubules, but for now just assume that it gets dispersed into the synaptic cleft in a reasonable manner, enough to reach the other side. When it does reach the other side, it's accepted by the receptors on the postsynaptic neuron, and essentially a graded potential occurs on the other side. This graded potential, whether it's excitatory or inhibitory, is dependent on the neurotransmitter. Again, GABA is the most major inhibitory neurotransmitter among other inhibitory ones, and glutamate is the major excitatory neurotransmitter. So if we get into so we'll say that the disbursement of the neurotransmitter is throughout the synaptic cleft. So how do we clean this up? How do we get rid of the excess in the synaptic cleft? You just let it float off into the extracellular environment and potentially disrupt other processes. Probably not a good idea in something as sensitive and delicate as the brain. So the brain has actually put a glial cell, a support cell here, one of the three support cells of the brain. This one is an astrocyte, which it takes up the excess neurotransmitter and does processes with them to make them more efficient. For more information on this tripartite synapse concept is what you want to look at, but it gets somewhat specific with enzymes and stuff. But once we empty the vesicles and everything's cleaned up, then what happens to the empty vesicles? They detach from the membrane and they get refilled at the axon terminal locally. So now refer back to the entire process before we look at the complexities. So we have the Golgi apparatus packaging, we have the ribosomes in the endoplasmic reticulum manufacturing. Once the apparatus sends the vesicles down, the transport vesicles, which are these down the microtubules, they arrive at the axon terminal independently of the action potential. The action potential, when the neuron is depolarized, it travels down, activates the Ca2+ ion channels. That causes the vesicles to adhere to the presynaptic membrane, releasing neurotransmitter via exocytosis, and the neurotransmitter binds to certain receptors on the other side, causing a graded potential. All the intricacies of graded potentials apply here, and we'll say the most important part, in my opinion, is that the distance of the dendrite, which is what this is, the dendritic spine, from the soma of the other neuron influences the effect of this input on the integration, the summation that the neuron does. So if it's close to the soma, like the soma's right here, then it'll have a pretty good effect despite being graded. If it's very far away, it will have a very minimal effect, but it will still have an effect. So that's an overview. Now we look at some of the issues here, especially in the microtubules. So these are important, once again, not for transmitting action potentials, but for the transmission of transport vesicles down to the presynaptic membrane. If these get messed up, like if we were to do, the vesicles come in like that, and then it gets crossed over the other side, gets crossed over like that, and it just crosses over here or something like that, and they get tangled up. Why doesn't this happen normally? They kind of remain in a nice structure. They don't interfere, and they are individual, and they allow things to travel down them because of this individuality. They don't interfere or do anything because there's structural integrity is maintained by a protein called tau, the tau protein. And this keeps them individual, this keeps them structured and consistent throughout the axon, no matter how long it is. In the case of a neurodegenerative disease, these tau proteins will dysfunction, the tau proteins specifically, and they will cut off into segments and aggregate together, forming something called a neurofibrillary tangle, NFT, and it will be accompanied by amyloid plaque. We can't see this in live patients as far as I'm aware. It's something that you have to cut the brain into many small pieces and check under a microscope that, yeah, this patient, he's now dead, but he has the presence of amyloid plaque and neurofibrillary tangles, confirming what we already knew, his diagnosis. But research is being done now to try and mitigate and potentially even repair these NFTs and repair the tau protein function. That's an interesting part about this. So when the vesicles cannot travel down the microtubules and attach to the membrane, then despite an action potential and despite the inflow of calcium, despite the adherence of vesicles to the membrane, there's no neurotransmitter, right? There might not even be vesicles here to do, so there'll be massive catastrophic dysfunction, and the Golgi apparatus and ER will continue manufacturing and packaging neurotransmitter, but where will that go? It will just create dysfunction across the board, extracellular waste inside the neuron causing oxidative stress, whole host of problems. And the other specificity is with the release of neurotransmitter into the cleft. Now in 2016 we had a research paper released, not me, but it was a very famous research paper that discovered the existence of something called a transsynaptic nanocolumn. Not just one, but multiple, dependent on which neurotransmitter the neuron was transmitting. If it was GABAergic or dopaminergic, etc., then the neurotransmitter would actually bind to a specific place on the membrane where a column specific to that neurotransmitter was present. And that column would directly channel the neurotransmitter towards the neurotransmitter specific receptor on the other side, on the postsynaptic neuron. This is huge, right? This adds a whole new layer of structure and order to the synapse connection. Now you might think, if there's a column, then how is it possible for these to disperse? If it's just going straight through like that, then why would there be neurotransmitter flying around in different places? It's actually such that when in periods of high frequency, these neurotransmitter columns will actually overflow, right? And then neurotransmitter will slip out of these crevices and escape. So the glial cell, the astrocyte, is responsible for cleanup of that, and that's an amazing efficiency, showcasing the effectiveness of the human brain at cleaning and recycling and being amazing, essentially. That's about it for axonic transmission. For further information, look up graded potentials if you don't know them. There is a difference between graded potentials and action potentials. It's small but very important. This is a dendritic tree. You might want to learn about how dendritic spines work and how their protrusions become dendritic trees. And also, it's beyond both of those concepts will be the concept, very, very interesting concept of long-term potentiation and long-term depression. This, on a very simple way, would increase the strength of a connection or decrease the strength of a connection through multiple processes. Yeah, that's it. But Yeah.